In today's lecture, we will talk about the enzymes. And the points that we are going to discuss in our this lecture are the definition of enzymes, the chemistry of enzymes, the classification of enzymes, the identification and acronym to remember the classification of enzyme. So let's start from the very first point, definition of enzyme. How will we define enzymes? We define in different ways. I'm going to tell you right now the very normal and the very general way that will cover your all dimensions. Enzymes are actually the biological molecules that act as a catalyst in a chemical reaction. Very simple. These are actually the biological molecules present inside the living body. And they are acting as catalyst in a chemical reaction. Now the question rises, what is catalyst? Catalyst is actually that thing which is going to accelerate a chemical reaction. In here we are talking about the biological reaction. So how will it accelerate a biological or a chemical reaction? Very simple. Let us clear through example. Suppose we have two reactions, R1, R2. They're going to react and produce a product. Now we will study this reaction in two ways. The very first way is that without enzyme. The very second way is that in the presence of enzyme. First, when you react these two reactants, here I have mentioned substrate. Now these two reactants in biological terms or in biological terminologies, when we are talking about the enzymes, then these two reactants are named by another term, that is substrate. You can call it as substrate or reactants, but I suggest you, when you're talking about the enzyme, you are supposed to call these reactants as substrates. So when these two substrates react together and produce a product without the enzyme and in the presence of enzyme. First, without the enzyme, when they react together, they will make a product, but the time consumed will be more as compared to in the presence of enzyme. Suppose the time consumed will be here about 7 minutes. Just an example. And if we react these two substrate in the presence of enzyme, then what will happen? Then the product will be formed within 1 minute. Just an example, okay? So now what happened? Before, when these two reactants were reacting, or these two substrates were reacting, they were producing the product. The product was produced within 7 minutes when there was no enzyme. And when we added enzyme, then the reaction accomplished within 1 minute. This is the reason behind we are calling enzyme as biological catalyst, because they are speeding the chemical reaction. When we added the enzyme, they speeded the chemical reaction. That's the very reason behind we are calling the enzyme is catalyst. Biological catalyst, okay? Biological catalyst. Remember. Let's come towards the next point, that is the chemistry of enzymes. Regarding the chemistry point of view, we have two types of enzymes, protein made and non-protein made. And remember, our most of the enzymes are protein made. And there is exception that we have, that is ribozymes, which is non-protein, means it is made up of nucleic acid, that is RNA. And you know what is the composition of RNA? That is made up of ribose sugar, nitrogenous base and phosphate. These three units combined together, they are making the RNA. Well, so we have exception, that is ribozyme, and the rest of the remaining, most of the enzymes are protein in nature. And you know, protein is made up of what? Amino acid. And you know the chemical structure of amino acid, any molecule that is composing or that composes the acid group or amino group in its structure, that is known as amino acid. Molecule is known as amino acid because it is having amino and acid functional group. So you know, when amino acids they chain up, I mean when their chain increases, then it is going to form peptide, then protein. Well, so we have enzymes of two, one is protein made, another one is non-protein made. And remember again, our most of the enzymes are protein made. Now let's come towards the next point, that is the classification of enzymes. According to International Union of Biochemistry, the enzymes are classified into 6 major classes. The very first one is oxidoreductase. These are the enzymes that are going to do redox reactions. Whenever you see any kind of oxidation reduction reaction in a chemical reaction, you are supposed to remember that this particular reaction is accomplished or is completed or is brought about by an enzyme that is from the oxidoreductase class. An example is dehydrogenase. We do have number of examples of these oxidoreductase enzymes, but I'm just mentioning one in order to give you just example of that particular class. Well, we have another one is transferase. whenever you see a kind of functional group transfer in a chemical reaction, that is actually done by an enzyme from the class transferase. Example is hexokinase. And hydrolase, whenever you see water added to any kind of reactant or any kind of chemical reaction, and that reaction undergoes a process of hydrolysis, then that is actually accomplished by an enzyme from the hydrolase. Example is lipase. Again, remember, we do have number of enzymes of each class. Okay, next one we have is lyase. If you see addition, removal of the carbon dioxide gas, water, ammonia, etc., these kind of reactions of addition and removal, when you observe in any chemical reaction, remember that this kind of reaction is being brought about by an enzyme from the class of lyase. Example is aldolase and etc. Isomerase, and if you observe isomerism in any kind of chemical reaction, remember that is from isomerase class. Example is phosphohexose isomerase and etc. Ligase, whenever you observe synthetic reactions, when two things or many things are combined together and they are going to produce another product, so there's such kind of synthetic reactions, when you see, remember that that kind of reactions are brought about by enzyme from the ligase class. An example is glutamic synthetase. Remember, again, we have number of examples for each class. Here I just mentioned one of each. Now let's come towards the next point, identification and acronym. Identification, approximately everyone knows that whenever you see ASE at the end of the term, that is going to be actually indicating the enzyme. I'm talking about biological terms, okay? Next one we have is ACR, that is acronym in short. So now this acronym is going to help you people in order to remember or memorize the classification of enzymes. Now what is the acronym? Just take one letter of each term. O from the oxidoreductase, T, H, L, I, L. Now just combine them together, make one word, that is othlil. O-T-H-L-I-L, othlil. Now this is going to make you people memorize this entire classification. O oxidoreductase, T transferase, H hydrolase, L lyase, I isomerase, L ligase. That's it. The acronym in order to remember the classification of enzymes. Hope so you got, and if still you have any kind of question regarding the entire topic, or if you have any kind of confusion, you are supposed to drop that in the comment box and we'll come for the answers very soon. Thank you for watching.